we continue our discussion on the digestive system of the human body by looking at the stomach. Okay, so the stomach, it's a muscular organ which can be enlarged depending on the amount of food material consumed by the animal. And in terms of function, it's a temporary storage for food mixing with digestive juices to facilitate chemical breakdown, to facilitate chemical digestion. There's several parts of the stomach. So generally, we have or uh, as a representation, so we have this picture here. So this tube or this sac is very muscular in material, and it has several identified parts. So we have the cardiac end of which is the anterior portion, the anterior end of the stomach contains the sphincter, the cardiac sphincter, which regulates the passage of food into the stomach. It allows food to enter the stomach and prevents it to go back or regurgitate to the esophagus. And then there is the fundic region, the body area, the body region of the stomach. It's an expanded part found only in the mammals, primarily facilitating temporary storage. And then the pyloric part is the end portion, the post-terminal part of the stomach with sphincter, similar with the cardiac end, and and it controls movement of food outside or out of the stomach and into the small intestine. Okay, um, the stomach functions limitedly in terms of absorption, especially of materials such as electrolytes, certain drugs, and alcohol. No, and its efficiency in absorption is brought about by folds called the rugae. So here you can see folds called rugae. So these folds here are actually absorptive sites. No, and stomach is modified across different vertebrates. In aves, in birds, the stomach is divided into two parts. There is the proventricular region and the ventricular or the gizzard region. The proventricular region, it's continuous with the esophagus of the bird and contains gastric glands, contains the juices, the digestive juices. While the gizzard is more of a muscular sac containing sand or small stones that actually help in grinding, crushing food material further. So in mammals, okay, especially in ruminants, okay, the stomach is composed of four chambers. There is the rumen, which is the region of the stomach where the plant-based food is fermented initially by bacteria. There is the reticulum, which is a compartment with um, which is a compartment that facilitates the molding, the clumping together of fermented plant material into cuds or small boluses. The bolus is regurgitated, masticated, and rechewed into the mouth. It is re-swallowed and then will pass to the next part of the ruminant stomach, which is the omasum. The omasum is a temporary holding space for the regurgitated food, and the abomasum is the true glandular stomach. Contains simple gastric glands for the secretion of digestive juices for digestion. Okay, so here more on the stomach. So if we look into the layers, the muscular layers that comprise the or that surround the or that define the vicinity of the stomach. So there is a longitudinal muscular layer parallel to the length or parallel to the direction of the gut. Okay, so here it's a longitudinal muscle layer. You see the orientations of the muscle fibers. It's longitudinal. And then there is a circular muscle layer surrounds the entire stomach. Okay. And there are mucosal and submucosal layers where other tissue types are found. Your usual epithelial tissues, your glandular tissues that secrete the digestive juices. The rugae, let's take a look at later at the appearance of it. No, so at the mucosal submucosal parts, what do we expect, or what is the function or the associated structure? So we have um, glandular structures producing gastric juice about 2 to 3 liters per day. And some important elements of the gastric juice include the following that are identified. So we have mucin, gastrin, intrinsic factor, pepsinogen, HCl, which is very acidic, pepsin, and gastric lipases. As well, the pepsin and gastric lipases are enzymes that allow for digestion of food material, while the other components allow for chemical breakdown. Like for the case of mucin, it's mostly a component of mucus secretions, no? Primarily made of carbohydrates and proteins. On the other hand, we have gastrin, which is essentially a hormone. It's a hormone produced by G cells associated with the lining of the stomach. Okay, so gastrin will, as a, in terms of function, gastrin facilitates or activates the release of hydrochloric acid. Okay, and then we have intrinsic factor, which is important uh, in terms of it's important in terms of absorption of material of simple molecules like vitamins. Vitamin B12 can be absorbed with the presence of the IF, or the intrinsic factor. And then of course, we have pepsinogen, which is primarily important for breaking down protein-based material during chemical digestion. Then, in terms of the structure, so here, the mucosal and the submucosal layers, we are seeing the glandular structures. Okay, so the cells that produce the tissues that produce or the glands that produce the gastric juices, we call them the gastric gland. Okay, it's primarily made up of several parts, several substructures, and then the gastric juice is released to out of the gastric pits. No? So here, right. So what are the specific functions of some of these cells? We have chief cells that facilitate the production of lipases, parietal cells facilitating intrinsic factor secretion, and then we have pepsinogen being converted to pepsin, no, in the presence of hydrochloric acid. Pepsin is the active component or the active form of pepsinogen that is carrying out protein breakdown. Okay. So here you can see an illustration on how what happens as the food material exits the and enters and exits the stomach. Right, so for absorption, so you can appreciate here the presence of rugae, the folds within the stomach that can allow absorption of limited material, limited types of material. So important things or points to remember. So in the stomach, physical digestion occurs via churning. So yung the movement of the physical movement of the stomach actually allows for food to be physically digested, and chemical digestion is facilitated via the presence of gastric juices. There's limited absorption happening in it because mostly absorption takes place in the small intestine. And then food in this area, or food in the stomach, is now called the chyme. Okay, now we'll look into the small intestine and the accessory glands. Right, so the small intestine is divided into three sections. Okay, it's the longest part of the animal digestive gut. Okay, and its length actually varies across animal groups. Usually, it's very long and mostly coiled, especially in herbivores. Usually shorter in carnivores and medium size for omnivores. So parts include the duodenum, which is the anterior portion next to the stomach, the jejunum, which is a coiled portion middle of the entire organ, middle section, and the ileum, which is the posterior, longer portion. It's the remainder and less coiled portion of the small intestine that follows the jejunum. So the inner walls of the small intestine are actually covered by a number of, or millions of finger-like projections called villi, which allow digestion and absorption of nutrients. So each vilus is additionally bordered with a short, delicate process known as a microvilli. So within the villi, there are smaller subsets of villi we call microvilli, forming the brush border appearance of the tissue sections of the internal lining of small intestine. And transport of material from the small intestine will follow, and or will transport of material from small intestine will be received by the large intestine. Okay. So let's take a look at one accessory, or at two accessory organs associated with the small intestine. So we have here our pancreas, and we have here the pancreas and the gallbladder. So for the pancreas, produces pancreatic juice. The yellow colored structure here is the pancreas. So pancreas produces pancreatic juice, and uh, um, what do you call this? Directs the pancreatic juice to the duodenal section of the small intestine. The pancreatic juice contains a number of enzymes for digestion. Some of these enzymes include, do we have, I'm not sure if we have that table, but anyway, so some of these contents of the pancreatic juice include proteases for breaking down protein, lipases for emulsifying fat, breaking down fat, amylases for breaking down starch, nucleases for breaking down DNA and RNA, and carboxypeptidases for peptide and amino acid um, production, yeah, for not necessarily production but extraction from your larger complex food material. Okay. I don't have it here. Okay. Let's see. And then we have the gallbladder, okay, which is associated still with our small intestine, the duodenal region of the small intestine. So you can see, there is a common duct that combines the secretions of the pancreas and uh, what they call this, um, pancreas and the gallbladder. The gallbladder primarily functions to store a liquid, for a liquid called bile. So bile is important for fat digestion, for fat breaking down, or no, fat chemical breakdown. So it's a mixture of different material, cholesterol, bilirubin, and bile salts. So the gallbladder primarily functions as an accessory organ that produces um, that stores bile. So when if the food material consumed by the animal is fat heavy, or it's mostly fat, it will secrete the bile. It will secrete bile in order to help the breakdown of fat. Okay. So furthermore, um, aside from storing bile, no, it also concentrates it. So the gallbladder also concentrates bile. What produces bile is another accessory gland, which is the liver. Okay. Now looking into uh, the function, the structure of the animal liver. So the liver, it's the largest gland, second largest organ of the animal body, secretes bile, which is alkaline in nature, about 800 to 1,000 ml of bile per day is produced by the liver. And I already mentioned that the bile is important for fat breakdown, no? Um, for the emulsification of fat, or I mean, in addition to fat emulsification, liver has several functions. So liver also include or has associated functions with lipid metabolism, with storage of nutrients such as glucose, no? The destruction of damaged RBCs can also serve as a reservoir for blood. Okay. So this one is the process of fat emulsification. So large fat globules are broken down into suspension of smaller fat droplets via the action of the bile salts within the bile. So the fat droplets become smaller and smaller as they encounter bile salts from the secreted bile. Okay, so here is a more visual presentation. So the fat globule, the bile salt serve as material that can break down fat globule into smaller droplets by having a polar and non-polar region, and the non-polar region primarily act as a scaffold to fat droplets. So fat droplets can be produced into smaller sizes from the fat globule. Okay. So here is a condition, no, where there is a problem with uh, um, what they call this. There's a problem with um, the breakdown of bilirubin, no. So the excess production of bilirubin and its unnatural decomposition or usage uh, can result to a condition called jaundice. No, so jaundice, um, also called icterus. Okay, it's due to high levels of bilirubin in the animal body. Okay, uh, usually there jaundice as it manifests is just a symptom. It can be associated with different types of conditions, no, but essentially the point is, or the main etiology is, there's excess levels of bilirubin in the animal body. So it makes the whites or the sclera of the eyes yellowish in color. Okay. Okay, so we mentioned this already. Okay, so the pancreas as a dual organ, right? So aside from secreting pancreatic juice that aids in the digestion happening in the small intestine, okay, pancreas also has other components or other structures that actually secrete material important for other bodily processes. So one of these structures are the islets of Langerhans, which secretes hormones into blood vessels. So the pinkish, excuse me, so you can see the pinkish colored spheres are islets of Langerhans that are with associated blood vessels. So these regions of the pancreatic organ, the pancreas, contain hormone producing cells. Okay, so these pancreatic islets are primarily function to produce insulin and glucagon for the metabolism of blood glucose, for the regulation of the blood glucose level. Okay. Now, in summary, we have this table here, which describes some which is a summary of the components of the pancreatic juice, of different enzymes, what substrates they act on, and the products, the byproducts of these enzyme actions against the specific substrate. So please make sure to review the byproducts emerging from substrate catabolism of specific pancreatic juice enzymes. Okay. Yeah, so here I mentioned a while ago about villi, and then the microvilli that are found within each villus. Okay, so here you can see the intestinal surface area, which is um, uh, possessing villi, okay, and the associated structures surrounding it. Okay, then if you look into each villi, and you'll see microvilli, which are actually increase the surface area for absorption. So as an absorptive cell, this brush border appearance by the microvilli actually provides more surface area than what is really available, or what is really physically seen, okay, on a gross observation. The microvilli associated with each villus increases the surface area, no, of the absorptive cells, allowing for efficient absorption of broken down chemical nutrients. Okay. And also, for the case of the brush border enzymes, uh, what are those, and what substrates they work on, okay, or they act on, and the byproducts of their activity. So please make sure that you'll take a look into this table, and have a review of them, or make sure that you review this table. Okay. Right. So, for as food passes through the small intestine, there are several mechanisms that take place. Okay, and there is what we call rhythmic segmentation and propulsive peristalsis. So, rhythmic segmentation are contractions that are most vigorous in the duodenal section in order to produce complete mixing of the food material of the chyme with bile and pancreatic juice, because essentially the duodenal area is the receptacle of the chyme, or the chyme from the stomach. So there should be enough mixing, or complete mixing of the chyme with pancreatic and pancreatic juice and bile, no? The resulting chemical digestion will break down food into its simple constituents. So these contractions, about 18 to 20 steps or repetitions per minute, are important for complete mixing. Propulsive peristalsis, on the other hand, the products of digestion are then propelled from the first segment of the small intestine down to the mid and the last sections of it, and reabsorption of the food material will take place, transferring nutrients to the bloodstream and the lymphatic system. Okay, so propulsive, it's section by section, unlike rhythmic segmentation where the entire column, or not column, the entire chyme is really mixed, or the entire bolus of chyme is mixed well with the pancreatic juice and the bile in order to maximize chemical breakdown. Then for absorption, of course, we have what things are, or what are absorbed. Definitely nutrients, okay, and water. About 10 liters of water enter our gut per day. And that's an estimate. If you drink a lot of water, that can be more, no? So the absorptive cells are the primary cells that facilitate the process of taking in the broken down material. Now, what are things that we should remember about the small intestine? It's a site where complete total chemical digestion takes place, primarily in the duodenal area, because of the presence of bile pancreatic enzymes and brush border enzymes. Okay, so bile is produced by the liver. It's concentrated by the gallbladder. Pancreatic juice, and the enzymes associated in it are produced by the pancreas. Brush border enzymes are, of course, found in the intestinal juice associated with absorptive enzymes. Physical digestion takes place via rhythmic segmentation and propulsive peristalsis, and increased absorption takes place because of the presence of the villi and microvilli structures. Food in this area at the small intestine is still called the chyme. Okay? Okay, now the ileum will empty its contents into the large intestine. So let's take a look at the large intestine. So, okay, so the large intestine, as um, a connection, or as a continuation, or the section where the small intestine exits, or gives in its digested material. It's the posterior portion of the intestinal segments. It's larger in diameter than the small intestine, so it's bigger, but it is shorter, definitely. Primary function is to reabsorb water, to dry out the waste or indigested food, and eliminate them as solid material or feces. No? It is the expanded portion of the digestive tube in humans and contains bacteria that can break down organic waste for the production of fecal material. And um, so essentially, it's you can liken it to a clean up, a cleaning, more of a cleaning section, or a cleaning unit of the animal digestive gut. Okay, so parts of the large intestine include the following. So we have the cecum, which is the junction between the small and the large intestine. In some mammals, it is large and it contains many microorganisms, many bacteria capable of digesting plant material, specifically cellulose based material. In humans, the cecum has an associated appendix. This one you can see here, the small structure. Then we have the colon, the ascending colon, ascending because the direction is going upward, and then the transverse colon, the descending colon, and the sigmoid colon. Okay, so the colon, it's the middle portion of the large intestine, and divided into four. Okay, so you can see here, ascending travels vertically to the right side of the abdomen, transverse travels horizontally, descending travels vertically on the left side, sigmoid is S-shaped and connects the descending column down to the rectum. Okay? The rectum is a posterior portion of posterior portion is also a posterior structure of the animal gut, and it's a temporary site for storage. It opens exteriorly as the anus, no? Both in insects, in mammals, and in other groups, in amphibians, in reptiles and birds. It opens exteriorly as the cloaca. So the anus or the cloaca is the posterior opening of a complete digestive tube. So essentially, its function is to, of course, facilitate defecation or the excretion of waste material. Okay. Uh, right. So, same processes happen. There's movement facilitated by churning and peristalsis in the large intestine, allowing for the transport of food material food material for reabsorption of water and for further breakdown to maximize food material acquisition or nutrient acquisition. Okay, so in the large intestine, some examples or specific bacteria that can be found. We have the bacteria Escherichia coli, which produces vitamin K for blood clotting. It's a fermentative bacteria for can break down carbohydrates and also produces several or makes use of several other nutrients or several other material for its growth and survival, which can be associated or can be acquired through the digested food. No? So the fecal material, which is released via the anus or the cloaca, is primarily water, 75% water and 25% solid material. Okay, usually contains bacteria, undigested plant material, because for the case of omnivores, no, and carnivores. Okay, if it happens that they get to eat plant-based material, it will not be fully digested. Okay, feces also contains fats, waste products, inorganic material, and dead cells from the intestinal lining. Okay, so some important things to remember about the large intestine. Chemical digestion is primarily via the presence of gut bacteria, physical digestion via churning and peristalsis, and water reabsorption takes place to solidify the waste undigested material. Okay, and to sum up the digestive enzymes, okay, so please take note of this important table. This will definitely come out in your final exam. Okay, and that's it. We complete our recorded lecture on the digestive system of the generalized animal body.